Okie doke. All right, Damon, I've invited you, so hopefully you can join. There you are.
Hey, yeah, sorry, it took a little while to
Hey,
figure that out.
no worries. Welcome. Everybody, meet Damon. He is one of the most fantastic mycological educators, naturalists, tinkerers. I have always been really impressed by everything he's put online, and I'm really excited I've gotten to know him and that we get to do this now. So, Damon, say hi to the folks.
Hey, everybody, nice to meet you. And thanks, Gordon. This is kind of fun to do this since we really can't do it in person anymore. It's what we gotta do these days. So,
Yep, I'd rather have you over for dinner. And the few times that Damon's been over, we busted out microscopes and got to work on looking all sorts of stuff. I think the time he came over and we were looking at morels, and then Jake
mhm.
showed up and we were sort of looking at cheese rinds, all sorts of stuff. I mean, it's amazing. You get a couple of nerdy people in a room together, and things just get interesting quickly.
I think just
Um,
about everything we ate that night went under the microscope at some point in time.
it's what you do. So, Damon, would you mind giving people and even refreshing me on a little bit of your sort of background and your story? Because you haven't come up necessarily through a traditional academic track, but I would say that you are as qualified, if not more qualified, than most PhD scientists that I know to be talking about mycology.
Well, there's not that many PhD mycologists, makes it easier.
True, but better than most PhDs I know. Period.
Yeah. So
So,
my background is more from like biology and biotech, and then mushrooms is something that came very very later on in life. So like if you want to go way back, I grew up like in actually I started in Oregon. I was born in K Falls, so any Oregonians probably know where that's at. Ended up in the Sierra foothills for my youth, which was really awesome in a lot of ways because you were just surrounded by nature. Ended up going to college, getting a degree in biology, and almost one in chemistry, and then ended up teaching a bit in high schools in Portland, and then coming back to the Bay Area to work on the Human Genome Project. So I ended up spending about 10 years of my career, more or less, in genomic sciences. So I was at a place called the Joint Genome Institute in Walnut Creek, California, which worked on 3 of the chromosomes from the human. And then after we finished that, the Department of Energy, who owned most of the facilities that were doing the Human Genome Project, was like, man, what do we do with you guys? We got all the sequencing capacity. We should have you, I don't know, biofuels. Let's do some biofuel stuff. And so actually that was maybe my first kind of gentle wandering into fungi because I'd
yeah,
never even like,
foray into fungi, as it were, right?
yeah, being a biology major, like it was like, one day in a class, like out of like a 4-year degree that you talked about fungi. And it was kind of like, well, just forget that entire giant kingdom branch,
Right.
whatever you want to call it. Just,
They're
yeah,
very much
forget about
an afterthought in any biological textbook. You have chapters on animals and anatomy and plants and photosynthesis and photosystems and all this biochemical stuff. And then sort of at the end of a chapter, we'll say, oh yeah, and fungi are important too. Look at how they help a plant grow. But beyond that, I was just reading some of Merlin Sheldrake's Entangled Life book earlier, and he was talking about, things are very animal centric, very plant centric, but we have very few people that are fungi centric, or at least who know how to put themselves in the footprint of a fungi and look out instead of always imposing their view on top of it. And so you had a chance to do that at JGI and actually profile some fungi, right?
Right, kind of get in there and understand like, oh, what are these things doing? Because like from the biofuel point of view, you want to figure out how to break down cellulose and lignin, right? If you're going to turn this into a fuel. And it turns out most of the organisms that we'd thought about, in like an undergrad biology, aren't the things that do that. It's mainly the fungi and bacteria that are doing that. And so a lot of time and effort went into basically figuring out these really large communities that were breaking down woody substrates, grassy substrates, how a cow's gut works, how a termite's gut works, and all of those things. And I kind of got my first little window into going like, geez, there's a lot there in this arena of fungi. And then what kind of helped solidify it at a different, more personal level was I went on a really long backpacking trip, the John Muir Trail. It's about 220 miles from
Wow.
Yosemite to the top of Mount Whitney. I'd never been on a backpacking trip over 2 days, and I was actually
That was
planning.
your first long one, was just go 250 miles just for the hell of it?
Yeah, well, and I was like the afterthought too. Like a friend had asked me, and then to just kind of come along because her and her other friend were going to do it, and then her other friend's knee got bummed, and I was like, okay, so I guess it's just you and me then. And being that long of a trip, and not having done something that long before, we didn't plan food resources appropriately.
Mm,
So we had been planned for 2 food drops. We got the first one, but it still had a lot of our food that we were carrying because we just weren't eating that much every day because we hadn't quite burned through all of our glycogen storage yet. And that happened probably 2 days after picking up that first food drop. So all of a sudden, like, we're really burning through food a lot faster than we thought. And then we eventually get to a point where we're going to be probably a day and a half, 2 days shy of having food from the next food drop. And at that point, like, your brain really kicks on and is like, what can I eat?
Yeah, yeah. I need
like,
calories.
Yeah, just like anything. And I remember like seeing like a Boletus edulis and like looking at it and just being like, I don't know. I don't even know. Like, I could eat this. It could be wonderful. I could eat this. It could kill me. I have no clue. And so when I got back home from that trip, we survived. We found berries and nuts and things. So we got the next food drop. I was good at that. I became very focused that year on just trying to figure out my local mushrooms. You know, partly because I thought like, well, I'd like to know them if I ever run out of food again. But then once I started kind of looking for like an edible one, I thought, okay, the first thing I'll go after is the chanterelle because I hear everybody in the Bay Area talk about it. It seems like it's easy to find. But in that process of just trying to find that first chanterelle, I just saw all these other things like fruiting that I'd never seen before because I never went with the intention to see things.
You know, I think that's a really good point because so many people are always asking me, you know, how do I get into mushroom foraging? How do I determine edible from toxic? And those are long answers. Those are not simple questions I can just give to people. And usually my best advice would be to find one edible species that you're fairly confident in IDing, and maybe you've had somebody show you how to find them, something like chanterelles, and go out and you can look for those. But ultimately, you're going to see a bunch of other stuff, and hopefully those things will engage you and keep you interested because you're never going to go out and just find chanterelles. And you're never going to, unless someone literally brings you to their chanterelle spot and allows you to pick all their chanterelles, you are never going to go out and get that lucky. You're going to be lucky to find a chanterelle, let alone, you know, a whole field of them. But, you know, what keeps you entertained is the fact that there's so many different things. And ideally, you get curious about all of those things. And that kind of cements a desire to learn more, which is, it sounds like what happened to you, right?
Yeah, because I realized in that process that like mushrooms were like the winter flowers for the San Francisco Bay Area. Because I go looking at plants and like, you know, flower season's a big thing to go be able to see stuff. And all of a sudden, like, I'm not sure how I missed that for that many years living here, right? And then all of a sudden you walk out and you're like, oh my God, there's all these things going on. How did I miss it for so long?
Once your eyes get opened, it's amazing how much is there. You just completely, you know, it's sort of like, I feel like the little agaric mushrooms you'll see fruiting on a lawn somewhere. That's like a perfect distillation of almost everybody thinks of that as a mushroom because we're all familiar with creminis and button mushrooms, and you see them on lawns, you're like, mushroom, don't care, doesn't matter. And then you start realizing the diversity of sizes and shapes and morphologies and all these other things, and you realize that fungi are all around you and you've just been missing them for years. It is this insane mind shift that once you become aware, you're like, oh my God, there's so much more life around me than I had ever anticipated. And some of it is edible, some of it is poisonous, and it's really confusing to try to figure out what's what, but that's where you start taking component by component and learning little bit by little bit. And then, you know, a couple of years later, you're a world famous naturalist with, you know, what do you rank? Are you still ranked in the top 10 on iNat? Because every time I look at it, I'm like, God damn, Damon, you know. Basically, for anyone who doesn't know, every time I go mushroom hunting, I will usually scope out an area on iNaturalist, because it's a great app. It's free. It's a citizen science program. And by the way, I want to talk about citizen science later. But iNat is a way you can kind of scope out an area. You look on like a Google Maps type interface, and it's like real life Pokemon Go. You can see all these places where people have taken a picture of something, tagged it, they've gotten an ID. Usually experts will comment, experts like Damon come on and help you figure out what's there. So I'll scope out an area, and I think probably like at least 95% of the time, if not 100% of the time, when I look at a new area and I'm like, oh, this is going to be a good one. I bet not that many people have been here. I'm like, oh, there's Damon. Damon's been here. And then I text you and say, hey, where the hell do I go? What am I looking for? But so I appreciate that. But Oh, sorry, go ahead.
I think it's really important because it helps people understand how much is around them that we don't see every day when you start engaging and trying to go and find stuff. And like iNaturalist is really nice because it gives people that stepping stone of like, okay, I can kind of figure some of this stuff out. And then also it just makes you realize like how much freaking stuff is all around you all the time that you're missing. and that we don't know. Like I just got a messenger thing today from a researcher named Danny Halewaters, and he was like, "Oh hey, how did you collect that fly you sent me a couple years ago?" And I was like, you know, I mean, he's expecting like an entomology like response, right? Like, oh, I had these like specific traps, and I was like, well, it was buzzing around in my apartment, so I swatted it with my hand, and I actually hit it, which is weird for hitting a fly usually, right? So I was like, oh, I'll go look at it, and it had fungal growths on it. And so I took a picture of it, you know, put it on iNaturalist, and then also actually used like a fold scope to actually look at the fungal fruiting bodies on this fly. And then this guy contacts me from iNaturalist, an academic researcher in mycology, and says, can you send that to me? And Danny just told me today that it's getting written up into a new, like, it looks like it'll be part of a new species clade.
That's so cool.
And like that's literally like, it's a fly in an apartment in a super urban area of downtown Oakland. Like how much other stuff are we missing just every day, right? And it just takes that desire to kind of see and realize that there's stuff around you all the time.
Right, and that's, I was going to say that's a great point for anyone who doesn't follow Damon already. You should totally go follow Damon, because although a lot of what he posts is stories and pictures from around Oakland, and particularly Lake Merritt, he is cataloging this insane biodiversity of a very urban area. And even the change in that biodiversity, especially with like a place like Lake Merritt, which has insane amounts of diversity, but not for a good reason, but because it's dealing with the international bilge waters that seeps in from the ports of Oakland and all of these invasive species and stuff that are out competing California natives. But what he's doing is keeping track of all of this and posting a lot of it on an app like iNaturalist, which allows other scientists and academics to look at that data and track it and make sense of it. And that's part of why I'm always recommending people use iNaturalist over one of the other apps for plant identification or mushroom identification that are out there, mostly because those other apps are scams and they're going to eventually try to charge you money. iNaturalist is funded by a Cal Academy, but it is connected to that citizen science ethos and background. And you've got guys like Damon on there that are, you know, superstars.
And iNaturalist too, like I feel like it's a better gateway drug than any of the other ones, because it's about all species, right?
Yeah.
So I mean, I actually started using iNaturalist mainly after I'd already gotten into mushrooms. So it's like, you know, majority of my observations were fungi. But then you like get down the slippery slope of like, oh, I want to know what this mushroom is, but oh man, I got to know what that tree is. I'll photograph that and get that. And then pretty soon you're like, oh, what's that moth on the tree? And then you're blowing an entire summer trying to figure out how to build like moth lamps like I did a couple years back. I mean, but what it does is it exposes you to what you don't know, right? And allows you to feel confident in getting to a place that you could know them, which is like, it's fantastic. In fact, if I was having to start learning mushrooms all over again, I think the first thing I would do is I would go to iNaturalist, and I could go out of season and I could just start browsing the area around me to see what's there. Because once you kind of understand what's there, you then can actually see it when you go there. It's all about expectation setting to some degree, right?
Yeah, totally. I mean, that's the kind of scouting that you can do is, you know, people are just like, where do I go? And I'm like, I really don't know. I usually just tell people, go to your local parks. But if you have a tool like iNat, you can at least scan and say, well, maybe there's no observations from my local park, but I know another place nearby that has a similar habitat. And you can look there and see what types of species they found. And then you have an expectation, a landmark for what you could be finding in your own personal park, or not personal, but, you know, in your own neighborhood kind of thing. So I guess I wanted to ask you, I mentioned citizen science, and I've had a couple people ask me, because I know I posted a story earlier in the week that said, hey, if you have any research or interest in academic mycology, or really just getting involved in science, and I think a lot of people do, how do you get involved in citizen science? Because I think you're one of the people who is a really good person to ask, because you've done lots and you know about some of the resources and the ways to get involved. How do people help scientists collect data and make good stuff happen?
I mean, I think the first thing is find something you're already passionate about doing. Like, don't think about this as like, man, I want to figure out how to be a citizen scientist. Think about like, what is something that I would be okay getting super nerdy on, you know, and like, I don't need anybody else's approval. I would just do this weird shit on my own. And that's where you want to start. And then once you get in there, start finding local people that are also interested. And what happens is you'll eventually just keep branching out, and you're going to eventually tap into academics that are also doing this. I mean, platforms like iNaturalist are really nice just because it provides that visibility. And now that a lot of academics have found it, like they're there mining stuff all the time. Like this relationship now I have with like Danny Halewaters is mainly because of iNaturalist. And I think I've sent him like, God, at least like 5 samples over the last couple of years. They're now in herbariums, and 2 of them are new species that are being described from urban downtown Oakland. You know, and it's like, but it's because I just had a flood of stuff on there, and I was observing and putting stuff up saying like, I don't know what this is. And it's that community process of having dialogue with other people that eventually ends up connecting you to people that can use that information. And so the biggest thing is like grow your community network about that thing you're passionate about, and eventually that'll have the ramifications where it connects to scientists. The other thing you could do is you could take that a different direction and be like, okay, I'm already really far down this pipe. Are there any big projects already going on? So for like mushrooms, like Mycoflora North America is a really easy one to get involved with, because there's an entire infrastructure of like how to document the fungi, how to package them up, how to even send them out to get DNA sequencing done on them, the whole 9 yards. And so there's some of these I call like, you know, citizen science in a box already. And so
you basically you get a kit in the mail, and you just go down the checklist, and then you're able to submit your own samples. And, you know, they're cataloged because you're following a checklist. They're cataloged in the ways they need to be for the scientists to add them to their collections, right?
Right. And then also like in this process of like working through the community and these 2 like groups, you learn what things are valuable and what things we don't know. And that then allows you to focus like, what do I want to work on? You know, which is kind of interesting because it's like, we all choose different paths. You know, like I look out there at some of these like non-academic mycologists, like, you know, like Jacob Polk, right? And he's like blowing stuff out of the water, doing like amazing new taxonomic work. And I'm like, that's really cool, but that's not my thing. Like I'm interested in doing a little bit of that, but like, I'm just as interested as like, when does this common mushroom that everybody sees every freaking day, Fruit Lake Merit? Because I want to be able to like associate that to weather patterns and things like that in my neighborhood. And so just find that thing that you're really nerdy about and kind of go off the deep end with it, but then document, right? Because that's the important thing. That's what connects that scientist and the community or citizen science, is having reliable data to go back to.
Absolutely. And I think that's a good point for anybody who wants to learn about the natural world. Is if you go out on a hike and you look at pretty stuff, it's great. But if you come home and talk to your friend who understands plants, and you're trying to describe this plant to them, you're like, well, it had green leaves, and, you know, there's some wrinkles on the leaf. Your friend is just going to go like this and be like, I don't know how to help you. I don't know what to say. And I get this a lot from people who send me, and I'm sure you get it too. People send you a picture of a mushroom and say, what is this? And you're like, well, that's like a top-down view of a brown mushroom in grass. I don't know, right? There's a thing in mycology called LBMs, little brown mushrooms. And you can't, like, there's some things I could immediately tell from the top. It's like a stinkhorn or something. It's very expressive. I can tell you what that is. But most things require some level of documentation, multiple angles, particular types of morphological characteristics that can help you reach an ID. And even then, sometimes stuff you can't ID from a photo. You're going to have to put it under a microscope, or you're going to need the actual type specimen in hand to do more analysis on it. But that documentation, you know, where you took the picture, when you took the picture, the context you were in, all of those things help as clues of identification. And, you know, to anyone who's interested in learning more about the natural world, you have a smartphone in your pocket. You can take almost all of the information you need at any moment and stream it to the world. So we have this incredible ability to be more connected than we've ever been before. We just need people to buy into it and start doing it. And we can learn so much about the natural world from this type of citizen science program. It's really cool. So.
Mm-hmm. And take time with the things that you find to like digest them in whatever way you digest it, right? Like when I go hang out with Christian Schwartz sometimes, it always blows me away. Is like, we'll be doing all much stuff, and then Christian will like bust out his notebook and then like sometimes sit down and like draw stuff, you know? And it's like a completely different way. When you draw something, you understand something very differently because you're looking at angles and things like that in order to translate them. So there's all these ways that if you just remember to also spend time with that thing you're interested in, you see things you don't normally see, which I think is also really important, right? Because like there's a lot that we like obviously don't know, and it just takes people focusing for us to kind of start seeing these new aspects of different organisms and things.
Yeah, I think that's a great point, because for me, getting into mushroom hunting, I'll be honest and frank and say that I was a pretty big wuss when it came to being outdoors. I was pretty scared of not scared, but dislike bugs, dislike being dirty. I was not a huge fan of hiking and getting all sweaty, and I just didn't understand what the appeal was. I was like, great, you can go out, and California's dry and deserty all the time. What are people looking for anyhow? And in the winter, like, it's raining. Why would I go outside? It's raining. Mushrooms shifted all of that for me and allowed me to be far more interested and engaged when I was outside, looking at the trees, looking at the plants, the soils, every aspect of where I was, because I suddenly had to pay so much more attention to find the kinds of mushrooms that I was driven to find. And when I found them, I was not just picking them up and looking for edible stuff. I was picking up everything, smelling it, touching it, feeling, and, just trying to understand what it was. And on top of that, I have a little macro lens, which you frequently have strapped to the top. There it is, macro lens. And I'm sure Damon and I will tell you that you can buy any of these little macro lenses. There's ones that are like 15x, or yours is a slightly higher X, but it's a smaller field of focus kind of thing.
Yeah.
They're cheap, and you can buy any brand. They all work fine. You don't need to ask us what brand. Just Google, strap-on macro lens. Make sure, though, it fits with the camera on your phone before you buy it. But looking at that gives so much more depth in terms of, oh man, there's features on this mushroom I wouldn't even have recognized. And I think when I first started taking a lot of photos, I wasn't fully aware what they were, but like looking at lots of stuff on Instagram, I was like, oh, that kind of mushroom has this cool kind of feature. So I started taking pictures of that, and then I started getting feedback from people hearing like, you're really good at taking these identifying photos because you identify these small features. And I'm like, oh, I just thought they looked cool from like artistic perspective, that there's reticulation or little bits of things here and there. And that's amazing once you really take the focus and go in, zoom in on things, how much more you can learn about them at sort of every level of biology. And that's, again, something I'm always impressed by. Damon does these incredible photo shoots with moths. He gets into it with mushrooms and sea creatures and plants and plant diseases and, all sorts of stuff. But I guess one of the things I wanted to go over with you while I've got you is bootstrap biology and mycotechnology. Because if you don't follow Damon, you may have missed the fact that he's done some really cool stuff. He's grown oyster mycelium on coffee grounds. He was like electrically stimulating mycelium. He's done all sorts of competition things and cultivated all sorts of wild mushrooms I don't think anyone's ever really tried to put in culture. So Damon, would you mind taking us through your home laboratory?
Sure, yeah, I'll do that. I might
and yeah, feel free to grab stuff as necessary.
Yeah. So what got me into this whole like home cultivation of like fungi was actually, it was some of the mushrooms I was trying to study in the Oakland Hills. They're too damn small in order to basically, for me at my limited knowledge, be able to identify them. So I was like, okay, I want to try and sequence them. So I work in biotech, right? So I know how to kind of do biotechy stuff. And so I brought them home to my basically home lab, which is, well, it's going to be my bathroom. So we're going to go in here. And here's my basically home lab for doing DNA work. And so I've got a thermal cycler, I've got a pipette, and I've got a gel box. And I basically can do gel extraction or DNA extraction here, amplification, and then run it out on a gel to make sure I've got the right size and send it out. So for some of these things that were really small in the Oakland Hills, I was finding I could just never get clean sequence. They would always be basically dirty looking, and I was like, well, what's going on? So I threw them on an auger plate, and really quickly what I realized was the reason that I couldn't sequence them is because they were also just full of things. And so like this would be like a great example. Like this is a mushroom from some wood chips around Lake Merritt that's having a hard time sequencing. And this was like 5 different cuts from it. And you can see that there's just a variety of things growing there.
You got molds and bacteria and other fungi and all sorts of stuff.
Yep. And so if you're trying to get DNA from that to then do sequencing, it's really difficult because you have too many different DNAs competing when you send it out. And so I basically was like, okay, I got to figure out how to do this cultivation thing. And I like doing things on the cheap because, I mean, I grew up in Calaveras County in California and just was not used to having resources. So I always am like curious about how do you bootstrap anything? I mean, like that home lab I just showed you there, it's going to sound like a high price tag to a lot of people, but that's about like $800 worth of equipment. But you also have to understand that in a typical laboratory space, that's probably more on the order of like $5,000 to $10,000 worth of equipment, a thermal cycler, some gel boxes, things like that, and even like DNA extraction. People are doing really fancy stuff. I literally am like taking like a pipette tip and then grinding the tissue and then using boiling and some other things in order to kind of separate materials. So real bootstrap. But in order to do cultivation, I basically have this setup, which doesn't look fancy. All it is a plastic
Tupperware bin
container.
turned upside down.
Exactly. And you'll see online, like, people show these and they, like, you know, cut holes in them and stuff so they can put their hands in. And, like, I don't know why you would do that, because then you can't use it as a storage box anymore. So all I do is I basically place these little boxes underneath it and prop it up, and then my hands go underneath, and then I can do all my work inside of there. And what I found was,
Oh, I was going to say that the point here of what Damon's doing with this little sterile box is by having a box over where you're working, things can no longer fall into your workspace, which is the biggest thing that, you know, contaminants and molds and bacteria and things will often fall out of the air onto your plates. So by having that, you know, above you, you have a somewhat sterile area as long as you spray the whole thing with alcohol and you're being careful with the airflow across the box and where you're working. But yeah, so go ahead, Damon.
Yep. And then, so yeah, so that's the space I'm using. And then I also was like, you know what, I don't really like using gloves all the time because you end up just making a considerable amount of waste. And so by, like I said, community, right, I learned that some of the things you could do that could minimize your chance of contamination and get rid of all of, like, the glove use is, like, literally just wash your hands really well with soap and water. And then what I do is I'll do a dip. Basically, I'll splash a little bit of hydrogen peroxide in my hands, wash it around. And so what that's going to do is that's going to oxidize a lot of things that might be on your hands, like the bacteria and things like that. And then after about 20 seconds, I'll move over and then just splash some isopropyl on my hands, and that will dehydrate anybody else. And what I found is I can go in there and it's pretty clean. I don't see any contamination from my hands. And I learned that from Sydney Oberstein, I think is his last name.
Yeah, Morcellophile. Yeah, Sydney is amazing. Super cool guy.
Amazing at this kind of cultivation stuff. And so it's that community learning again, right, that allows you to kind of do stuff.
Which again, a good point is that community, you can find that community here on Instagram. You know, this whole Fascinated by Fungi thing just came out of me having hundreds of photos on my phone and having no idea what to do with them. So I just started posting them. And, you know, a couple of years later, I've found this insane, incredible community of really smart people who are doing stuff like this that you can learn from on a daily basis. So,
yeah, it's really amazing. It's just, all you got to do is kind of tap in, right? And then ask questions. I mean, that's the biggest thing. There's a lot of, like, Instagram and then some of the Facebook groups. There's just people, like, willing to give information all the time, right? And it's not like you're just learning from a bunch of randos. You're learning from people that are, like, really at the kind of the top of their game. Like, I mean, the amount of, like, work that Alan Rockefeller does, like, in a digital platform for people to get access to learn stuff. Christian Schwartz, Sydney, I mean, all these folks, it's incredible. And so with that home cultivation, I'll do auger work. And what auger work basically means is I'll take a pair of tweezers. I will pull apart a mushroom I find to try to get to inner tissue, because that has a less chance of being contaminated, right? That outside stuff has got the whole forest on it and whatever floor my hands had when I picked it up. So if I can tear it open, pull out some tissue from the inside, I can then put it on a plate and it can get relatively clean.
Is there tissue that you select, like gill versus, like, you know, actual tissue, or is there some part of the mushroom that you kind of aim to isolate?
It's a good question. So it depends upon the size of your fruiting body, is what it really comes down to. So what I'll try and do is, if it's a small fruiting body, I can't be choosy. All I'm choosy about is where can I get a piece of tissue that's, you know, bigger than like a pinhead that isn't attached to the surface someplace. And sometimes that's a hard gig. But other things, like somebody recently, Nikki, gave me this really cool bolete that grows on dead Pinus radiata around here. I've always seen dry. I've never had a fresh one. And this guy from Instagram, who's like, I follow your account. Hey, would you try and get this in culture for me? I was like, oh, hell yeah. I'd love to get that in culture. And so on that one, you know, it's large enough I can open it up and I can take cap tissue that's really far away from everything. And then I could also take stem tissue. And so if I've got a large mushroom, those are usually the two I go after, is stem tissue and cap tissue. I usually stay away from the gills just because the gills can be exposed to the outside world. And so that's also, I mean, when you look at like a parasite attacking a mushroom, it's usually where they start, is like in those gill spaces, right? And so I'll usually start with inside cap tissue or inside stem tissue to keep things clean. And then usually what it takes is, you know, you start off and like your first plate will look like this. Like this is the inside tissue of that mushroom from Lake Merritt. And you have to kind of like baby this. You watch it every day, you check how it's growing, and then you're like, okay, I see something that doesn't look like mold. Now I'll transfer it to another plate and hopefully it gets clean. And then this is that same mushroom now completely clean. And so now I've
So you
got,
can, yeah, you can see that central point where you took some agar from the
yep,
previous plate, put it in, and the mycelium has expanded around, growing out in equal directions because that's on a solid media surface.
yep. And so agar,
Very
for you
cool.
guys that aren't aware, it's just basically like Jello but made from seaweed. And then this has malt extract in it, so the same stuff you make beer with. So as a former home brewer, this is a really convenient hobby because you already have all the gear here. But the crazy thing is that some things don't always grow like that one that you see. So sometimes you get things on agar and all of a sudden they do things and you're like, wow, that's really beautiful. Like this is Armillaria, the
Oh
honey
my
mushroom. If
goodness.
you've ever seen honey mushrooms in the wild, they have these huge black rhizomorphs that will go through the wood. And if you put it on agar, it does that exact same thing. It starts growing out these like just funky giant rhizomorphs. It's just beautiful.
So for anyone who doesn't know, a rhizomorph is a form of mycelium that's basically been weaponized. It's hardened, weaponized mycelium. And Damon, correct me if I'm, going off the roads here. But
Mm-hmm.
Armillaria are the honey mushrooms, and they are super parasitic, sort of competitive mushrooms that will go out and with those weaponized rhizomorphs sneak up in between the bark and the tree, and they will girdle a tree, and they'll absorb all of the nutrients and stuff that are in the xylem and phloem and kill the tree, and then eat the tree. So they are insanely competitive. And you guys have probably heard of that giant patch of Armillaria that's up in the Pacific Northwest and spans like 3 or 4 states, kind of thing. That is ever expanding outwards with those gnarly. Can we see that plate one more time? Those
Yeah.
gnarly black, if you were to like, just the color of that plate with the black and sort of red contrast, what is some sort of demon spawn in your house look like? Oh, it's, if you ended up with a spot on you that looked like that, you'd be like, I have some evil that needs to be purged inside of me. And that's what's in the soil. That's that weaponized mycelium, those rhizomorphs. And it's interesting because some of what you're showing me here with these plates reminds me a lot of my background where I was doing native yeast isolation. So in my time at Davis, we were trying to profile what does a native California Saccharomyces yeast look like? Because most of the yeast that we're using for winemaking and brewing ends up being stuff that was imported over from Europe, or honestly, if you look at the history of domestic yeast, a lot of it ended up coming from China. And so a lot of the strains we have originated genetically in China, and yet they've been used in human wine and beer production for hundreds of years. So we were out trying to profile what did a native California yeast look like, and their habitat tends to be on the effluent of hardwoods. So we were going out to fairly remote areas, actually like the Pope Valley right now, which is completely on fire. But we were out there looking for trees and trying to find little wet areas on the trees and swabbing them, or taking a piece of tape and then putting that in a tube, sort of as aseptically as you could, pouring juice over, selecting for things that were fermenting. Clearly, if it didn't ferment and it molded over, we'd just throw it. But if it was fermenting, we would try to plate that out. And we would use there's another media we had called WLN, which is this sort of beautiful bluish green media. And yeast will turn the media yellow because they pump out a lot of acid, basically. It changed the pH of the media. But it was a similar process where we would have to go through like 4 or 5 rounds of plating, selection, plating, restreaking, and that's the purification. And that's sort of standard microbiology. So what you're doing is very much in the sense of like what microbiologists have always done. You're just doing it with mushrooms, which I think a lot of people haven't put the work into that with a lot of mushrooms. So it's really cool that you're doing this, especially in your own house. You're not being paid to do this. This is your hobby.
Yeah. I didn't know that about the yeast, that you guys were going out to hardwoods and doing that. That's really cool. But I can also see how much of a pain that would be, because you're getting probably a pretty big diversity if you're just doing a pull off of a hardwood wet spot.
It was selected for a little bit by the fact that they do fermentation, so they'd create alcohol. And that was enough to knock back a lot of the bacteria and some of the non-sac yeast. But yeah, it wasn't easy, and there was times when occasionally I'd use media that had a little bit of antibiotic or something just to help with the selection. But I mean, my sort of crowning achievement of that came when I was, after I finished grad school, I went and worked to harvest at Opus One. And I was supposed to do a lot of stuff there, but one of the things I was not supposed to be doing was working on the yeast. They had another guy for that. And I was like, well, I have a yeast PhD, so really that was a stupid move to bring me in if you didn't want me anywhere near the yeast. But one day, me and the other guy were going for a walk, and I told him about this whole sort of yeast isolation thing. He was like, well, I'm interested. Can you show me how it works? So I taped a couple of the random trees around this winery, and we ended up isolating a strain. I did some test ferments, and I showed the winemaker and was like, hey, can I get like a little keg of juice to kind of mess around and make wine with this? And he was like, you know what? I'm going to give you a 2-ton tank. And I was like, are you serious? This is your grapes are not cheap. This is a lot of money. And he's like, yeah, you can do a 2-ton tank. And then we went out to a big lunch, and we all had a couple bottles of wine and stuff like that. And after lunch, he comes to me and said, screw it. You're doing a 20-ton tank, not a 2-ton tank. And I was like, are you serious? So our crowning, and what I was doing there is I was taking this yeast from plates, scaling up to like 1 liter bottles, and then we were dumping it in these sort of bioreactor tanks we had, and I was growing up a significant amount of yeast. So I dumped all this into our 20-ton tank. We took a branch from the tree that it had been isolated from, and they had a bunch of sort of funny names for their house yeast. They had like the wolf and the owl and the coyote, these sort of epic animals that were around their vineyards. And because this one came off a tree, they named my yeast the squirrel, which I think is hilarious because now they have this like suite of epic animals plus the squirrel. And it's become one of their favorite yeasts. I know they make wine with it every year now. And so that's one of those things that I love that has happened. And, you do that kind of isolation and it can bring about, new things. And I'm sure there's, in all the work you're doing, you're going to be collecting mushrooms that people are going to be seeking out, wanting to do commercial cultivation with, or, getting enzymes from, or using for research, or, like you said, writing papers on a fly that you found in your apartment in Oakland. It's really mind blowing what you can find.
Yeah, that's really cool. I'm actually really impressed with that yeast work, Gordon, because I dabbled in that a little bit at Lake Merritt too for a while. Like I had a buddy who was starting a brewery in Lima, and he wanted to figure out how to go about getting wild yeast. And I'm like, I've never done it. Let me try and do it where I get everything from, my local urban park. So I went around and basically, I never thought about going to hardwoods, but I sampled all of the flowers with any sort of nectar in them. And man, just the diversity of flavors. I would just do little tiny, like,
mini
500 ferments. And wow, it was I was really blown away, especially because having been, a guy that brewed beer for a while, that so much of that profile was coming from the yeast, right?
Yeah,
like especially
totally.
like I remember getting one from an orange tree, and it was like spot on. When you open the jar, it smelled like the orange blossom, which completely like changed the way I thought about orange blossoms. And then when you tasted it, it had a lot more of that Hefeweizen aspect, right? Because it's wild and it hasn't lost those canola pathways and things like that. But yeah.
Well, it's funny that you're a home brewer, because I think what led me ultimately and circuitously to mushrooms was being a home brewer in college. And it, came out of, I liked drinking beer, as many, men in their early 20s do. And I
cheaper.
Yeah, well, exactly. You think it's going to save you money, and then it doesn't save you time or money. And ultimately the beer you probably ended up brewing wasn't as good as something you could have bought at the corner store. Because you have to worry about sanitation and ingredients and all these other things. So at some point I realized that I was not going to become the next greatest, brewer in America. But I did realize that I could learn more about yeast, which led me to grad school and working with my advisor, Linda Bassan, and I learned a ton about yeast genetics. And then that was coming out of grad school, having that background in sort of the genetics and the understanding of fungal lifestyles helped me just jump into and really run away with my passion for mushrooms. So it's amazing to hear too that you as a home brewer helped get you sort of started. But
well, it gives you a lot of tools, right? Because it's like the biggest thing from going from working in a lab space and then working at home is like figuring out how to be sterile at home. And I feel like home brewing is one of those things that gives you at least a leading edge on that. You kind of
definitely.
like realize, like, I mean, you guys can see my place, like, you don't need to have like a clean place, right? But you got to figure out how to be clean. Oh, actually, there we go. How to be clean in the space that you have for the things you're cultivating. And I think that's a really important learning piece for anybody that wants to do home cultivation of different fungi. And then once you do it, you get a real kind of knack for it. You can do things really quickly, and then you can really start to play and innovate, And I feel like I've finally, like, after like a year of doing this type of stuff in my house, like I finally feel like I'm at that level where like I know where my contaminations come from, and now I can really play, you know. So like, you know, for example, like here's like some oyster that I started, and I don't have a pressure cooker. So most everybody that does home cultivation has a pressure cooker. I don't have one. I could easily buy one. They're cheap.
I was gonna say, I'll order you one on Amazon right now.
Yeah. So I was like, I want to figure out how to bootstrap this, right? And so I looked around, and I remember like reading for home brewing this thing called the Tyndall sterilization technique. When this idea is that you would basically bring something up to a hot temperature, let it cool down, and then let it go for a couple days. Because what happens is any spores that survive the heat for the first time, which a ton of things can survive that first like hot cycle, will basically go like, ooh, it's safe. I've got a nice space now. I'll kind of come out, start growing, and then that's when you get them again with heat. You hit them again with a little bit of heat, and then you do that one more time. So it takes like me like, you know, 4 to 5 days to sterilize my stuff, but I don't need a pressure cooker, and I'm literally just doing it with heat and the ability to like close the system. Which I think it's very kind of powerful feeling, because this is then something you could translate to people that, you know, don't even have a stove, right? Maybe all they can make is a fire, but they could do mushroom cultivation just by being able to heat something up, which I think is kind of cool.
And
so
I was going to say, that's a great point with fungi, is that a lot of the times sterilization is sort of necessary to get started. But especially if you get like a really nice block of something that's well myceliated, you've given it a huge head start. And then you can introduce it to systems where there's a lot more diversity, because you've already got a really good starter culture. And that's like what I was doing with the yeast at the winery. Was we knew that the juice was full of other organisms, but that if we put a high bioload of something that had been grown up under optimal conditions into that ferment, that it would all go off. And similar kind of thing with your oysters, you could probably take one of those nicely myceliated blocks and drop that into substrate that wasn't sanitized necessarily, and watch those oysters still become the competitive, you know, main species there.
Yep.
So,
exactly.
I mean, what are you doing with those oysters? Because I also know that like you've experimented quite a bit with substrates,
right.
You know, sterile and non-sterile.
I mean, so one of my favorite like substrates is just like leftover food waste in my house, right? So like here's a sterilized jar of Tyndall sterilized of like my tea leaves and coffee, and you can see I inoculated it right there. So we've got little oyster starting right there, and I did some other points around it. So this thing will myceliate in probably about 2 weeks. And I literally can open that up and then just put it in like a little humid box. And now I'm getting oyster mushrooms from my leftover food scraps. And then that block, once I feel that it's done, that goes in the backyard, and that's like rebuilding some of the soil that we have like in the backyard here in Oakland.
Talk about sustainability there, man. It's you're not wasting anything.
And for oysters, because they grow so fast and easy, I feel like they're a really nice one to actually like play around with. So like recently, and actually this is after, you know, reading The Entangled Life that you're on right now, you know, he was going over some of, a beautiful book, like a great quick update on what's going on at the leading edge of kind of like the fungal community. So I read that, and I was kind of like, those are some really interesting ideas. I'm going to read some more about electricity and mushrooms. And that was like, you know, like one of those black hole nights where
a deep dive.
Oh God, it's 4 a.m. and I've got like a conference I'm supposed to give at 8 a.m. Oh no.
How many papers did you end up downloading? Because that's sometimes I look at my download folder, I'm like, why do I have like 30 papers in here, and like none of them, I don't have them organized anyway. This is just like clearly stream of consciousness where I was like, you know, putting in search terms and like, how much can I get?
Well, that's the danger, right? So like I got it at 4 a.m. check, and I was kind of like, okay, I think I'm going to digest all I can. What I should probably do is just right away start trying to play. And then I can always digest the papers as I'm playing and kind of getting a feel for the systems. And so my first attempt to play was I built this little guy, which is basically a petri dish with just, you know, 2 electrical leads running into it that I could then put a 9-volt battery on it. So then I could shock my mycelium. And so I basically dropped some oyster in there, and then just would shock it, you know, for about 5 minutes every day.
Okay. So this isn't continuous. You're just doing little bursts of electrical activity.
Well, because when you don't spend the time to set up an experiment correctly and just playing, you realize that when it's on there for a long time, it actually just starts to burn the media. And so then you're like, I will backtrack now and just do a little bit of time. And so that's what happened in this. In fact, there was a really cool, if anybody's into electrochemistry, there's some really cool copper oxides coming off of the positive terminal here. But then I was like, oh no, because copper's used a lot of times to basically prevent specific fungi from growing. I was
it's
like,
an antifungal.
It's going to bankrupt this. Oyster doesn't care. It just blew right through it.
Which you shouldn't be surprised by, because oysters are like the mushroom or the weeds of mushrooms. They will just grow through anything and super competitive.
Yeah. But what I saw was, and it's hard to see now because the whole thing's basically filled up, was I did see that the mycelium actually formed kind of stronger networks heading to both the anode and the cathode, which was like, to be honest, like I didn't know what to expect by doing this. But now I was like, oh man, now I feel like there's something there. So while I'm reading these other papers and digesting, I've got to move on to actually like setting up an appropriate experiment. And so I have done that. And actually that's running right now. So why don't we walk over to the other side of the room? So we'll go past the beer and we'll go over to here where I'm actually using more of like a sophisticated setup. I have 2 gel boxes running. And in each one of them, there is a plate with oyster in there. In this one, I burned holes in the Petri dish, so that way electrical current can come into the dish and influence the mycelium. And this one, which is my control, just basically doesn't have the burned holes, so it's just sitting in a field, but without having the actual electricity pulse through the gel.
Gotcha, okay.
And then with this, the beauty is I can actually control this a little bit more. And so I'm bringing it so the voltage is about 20 instead of 9, and then doing this over like a much longer period of time, which is kind of cool. Which anybody could actually do this at home without a fancy system by just building one of these. So this is this same system, what we call gel electrophoresis in biotech, just using some 9-volt batteries and literally like an old thumbtack box and some alligator clips. And what you can do is you can pour a gel in there of just like Knox gelatin and literally put it like, you know, if you wanted to like separate biomolecules, or I could just drop my Petri dish in here now with some tap water and just a touch of salt, and you'll have enough current that you'll be able to basically get a really strong field in there. Or you could use the same system to actually separate DNA. And so there's always, what people always forget is like, there's always a way to hack something, because every single scientific piece of equipment you see in a laboratory was at some point a completely rigged piece of craziness like that. And so you can always go and remake those. And the beauty is it doesn't cost much. You may lose a little bit in like, you know, like austerity and feel, but you can make it happen. And it actually helps you really understand what that device does by building it and using it in that way too.
The principles are that much more right in front of you when you see that you know, you look at that box and you assume there's electronics and all these other things. And then you look at literally a plastic dish with a couple batteries and a few leads beside it, and you realize that there's not that much more to it, right?
Yeah.
It is phenomenal. And it's funny because you work for one of those biotech companies that sells those very nice boxes where everything is compacted and streamlined and all that kind of stuff, but nothing helps you understand it more than getting into the nitty-gritty of it, taking away all the fluff and saying, here's the principle and here's how I can use it. So that's part of why I love following you, because I'm constantly learning from these experiments you have going on. I also saw that you posted a paper about, I think it was lightning strikes influencing and stimulating Matsutake mycelium, which was really cool because I'd heard that reference by Paul Stamets and I was a little skeptical, but when I saw the paper, I was like, oh, someone's actually done this. So, I mean, what were your thoughts on that?
I was blown away because I was like, what the hell are we doing in North America? Like the Japanese are literally building like a little like push cart that you can strike the ground with electricity so that they could run this experiment, you know? Like for us,
portable lightning, right?
Yeah. And I was just like, oh my God. So what they saw was that by doing this, and it was a really well-controlled study. So they had this area that was known to produce matsutake. They carved it up to a number of plots, and then they basically did the push cart on a couple of plots, didn't do the push cart on others, and then basically collected mushroom fruiting bodies for 2 years from there. And what they saw was like a 2-fold increase in the number of fruiting bodies and a 65% jump in the total like biomass that was produced as a fruiting body.
Wow.
So for all those people that were like, does lightning cause mushrooms? Well, apparently it might. And that kind of goes back to a little bit of this, which is fungi will respond to basically stimulus at one side of their mycelium all the way to the other much faster than can happen with chemistry, right? So instead of having a chemical signal, they must be doing something else. And so a lot of these researchers in the last couple of years have realized that some of these hyphal networks are actually acting very similar to how like nervous system cells will work, right? They actually can
They'll
run
send
an action potential.
action potential that travels down just like a neuron would of, you know, it's changing the calcium sodium balance inside the cell, and this action potential runs down
Right.
like it would like an axon or something like that.
Which is incredible. And then like, you know, some of these researchers, some of the papers I was reading, you know, they would expose it to different types of things, and you would get different basically action potential signals over time, which is like very obvious that there's like different types of coding going on. It's not just like our nervous cells, which sometimes are just like on or off, right? Like retract your arm or don't retract your arm type of thing, but like complicated amounts of information are getting moved around through these mycelial networks, which then opens up this whole other world, which is, A, can we start to learn what those signals mean? And then from a cultivator side,
is there a language that we could interpret? I mean, not even a language, but even just a series of systems, integration, and understanding those signals that we might be able to tap into. Imagine taking a rod, sticking that into the dirt, measuring amounts of action potentials or electric potentials, and inferring, you know, microbial diversity, health of the system, what the trees are doing, nutrient transfer, all that. And we're blind to it because we as human beings are like, mushrooms don't talk, they don't speak, they're not intelligent. And that's us trying to anthropomorphize them from our view. But, you know, again, what I spent a lot of time in grad school doing was, well, how do I put myself in the place of this single-celled fungi? How do I think like an organism that has no discernible intelligence and yet is incredibly adaptable and is everywhere, is ubiquitous? And I think it's a similar kind of challenge when we look at fungi, is how do we put ourselves in that place and completely change our worldview to understand, you know, what's beneath our feet and the glue that holds all the life together, you know? So
yeah. And I'd say, I know it's going to be interesting because like somebody will crack that code, like in our lifetime, right? We'll crack a code that allows us to understand at least what some of this communication is in very basic networks, right? And then hopefully we can scale up and get more complicated, right? Because I mean, like we can't even figure out how a C. elegans worm like works, and it only has 939 neurons, you know. But we'll get there, right? I mean, like when we worked on the Human Genome Project, people in the 90s were like, why are you guys like, this project will never be finished?
Insurmountable amounts of data. You couldn't possibly figure it out. And now, you know, we I don't know how many human genomes we sequence a day, but, you know, in another 10 years, 20 years, it's going to be a pretty standard part of your health protocol. You know, you'll go in, you'll get a cheek swab, they're going to send it out for sequencing, and you're going to have all of these alleles and this huge health report. And I mean, this was kind of already done with like companies like 23andMe, and unfortunately the FDA kind of tamped that down in terms of the effectiveness of what they're able to tell people. But personalized medicine in that sense is coming, as is like microbiome sequencing, all sorts of stuff that is really, you know, going to paradigm shift the way we look at health. And I think mushrooms are going to be a big part of that, because we're going to see, I think, more and more influence of mushrooms on medicine. And, you know, not just psilocybin, which has been very fetishized by, I think, certain parts of the mycology community or people outside of the mycology community, which is great. And I'm glad there's so much attention on it. But I think sometimes it takes away from the stuff that I find even more interesting, which is what you're doing, and, you know, the citizen science, the mycotechnology, these incredible things that are being done with materials and food and yeah, medicines and therapeutics. I mean, I don't know, is there anything out there that you has recently blown your mind from sort of mycotechnology standpoint?
Man, it's so hard because like after finishing Martin's book, right, I went all these tangents, so it's just kind of like all over the place. I mean, I think some of the material science stuff is really cool. So like, you know, companies that are rethinking how we get some of the materials we're used to having. So I mean, like the most obvious ones, if you're in the Bay Area, is like Bolt Threads or MicroWorks trying to make, you know, leather-like products, basically using Ganoderma, growing Ganoderma. But that's going to go much further than that. That's just like, you know, I mean, when did humans start to tan animal hides, right? We're basically just at that point with mushrooms.
Yeah.
But then ultimately what we do is we learned how to like take those types of materials and do radically different things with them. So at some point, you know, growing hyphae to then ultimately use it as maybe a way to transmit signals. Or maybe then, you know, replacing the hyphae with something else by doing like a lithography So that way you're able to kind of grow networks, things like that. And I think they're just like, we're really just at the starting point of understanding the material science potential of using mushrooms. And I think on the medicinal side, it's a lot harder of a road just because it's so clouded with all of like the hype stuff, right?
Yes, yes.
And that it's like a lot harder for people to weed through, where like material science doesn't have that. Nobody's been saying you should take a mushroom and wear it. Well, people were doing that. Okay, you can take a mushroom and turn it into a computer, or use it to grow your chairs, or your yada yada. But with medicine, it's been a lot different. You have like a lot of like this kind of like urban shamanism that's developed and fetishized a bunch of these mushrooms that there's some truth to it, but then there ends up being a lot of like hand waving.
There is, and I try to express that to people. And I remember I've given a couple talks on it, and I've had people get very offended. And so I try to be pretty careful when I talk about it, because I don't like to shift or challenge people's worldview in a way that they find offensive. But there is a lot of sort of snake oil that goes along with some of those mushroom supplements, because there's no clinical benefit necessarily to taking those extracts or those supplements. And I think the thing that a lot of people miss is that there are bioactive compounds in mushrooms, definitely. And there is, biochemical proof of that. And there's biochemical proof of those compounds potentially having activity in an in vitro system. But is taking a mushroom pill for your whole body going to actually actively fight that cancer? And then how much of that bioactive compound is in that pill? How much of it is going to get to that particular tissue? And that's what we just are lacking evidence on. And it's not, you would take that medicine when you're sick, when you have something that you want to cure or work on. It's not something that you need to take every single day as sort of a supplement. And I think one of the things I also express to people is, if you're, instead of spending $50 on a little bottle of tincture, go and spend $50 on mushrooms. You're going to get a lot more food, and you can have some people over for dinner and probably have a great interaction that will help your mental health. And much like juicing, having the actual mushroom with all of the fiber in it you're going to get a lot more out of that and digesting that than you would out of, an extract of it. So.
Yeah, I feel the same way. And a lot of times I'll take it even one step further, which is like, go out and try and find it. Like, take that group of people and be like, let's go for a walk, and maybe we'll get to add some mushrooms to our meal tonight. And that connects you to your land. You also see things in seasonality, which I think is something that we forget a lot about in diet. People are like, I'm going to take this reishi extract every day. And it's like, even like if you're trying to do this, like, I want to think about how humans usually were back in the day. Like, well, they weren't taking something 365 days a year. They're like, oh my God, I found this thing for a couple days. I ate a whole bunch of it, And so I think that's something that needs to be kind of, thought about when people get too kind of caught up in the medicinal aspects of it, is that there's a lot of benefit from the mushroom itself, the seasonality of it, etc.
Right. And it connects you to those natural rhythms of the world that, again, makes you sort of a better citizen. When you go out in nature and you look for these things, you're that much more connected to the seasons and the bugs and the trees and all that stuff that you can take photos of on your phone and put them on iNaturalist. Or you can send pictures to us, and we will get confounded by what you're finding, and usually probably ask you to send a better photo.
But also it gets you to appreciate that land that's around you, understand how it's changing, how it's not changing, what different seasons look like. I mean, and you also just like it almost gives you like a spirituality with the place around you, because all of a sudden you realize that it's not just this static thing. It's constantly changing every day, and it just takes you going out and looking at it to make you realize that. And then you think like, wow, how are those things affecting me then? Because all this is going on just right in my general area. It also gives you a much greater appreciation for like just the space around you.
We're going to run out of time, but Damon, it's been so much fun. Thank you so much. And thank you, everybody.